hard way too, because then you get nervous when that's Let me just double check. Valerie, I just thought about it. Did you get a photocopy of her stuff?
Hi everybody, welcome to the last round of the lightning talks of the semester. My name is Hughes, and make sure you have a beer or a wine to make the presentations more enjoyable. The first presenter is my advisor, Franziska Michor, and please go ahead.
Thank you, Ken. So I want to show you what we are working on right now. It's something pretty different from what we usually do, but we've become interested in DNA methylation profiling for non-invasive early detection. And so the problem is obvious, right? If we were just able to detect cancers earlier, then we could come up with a surgical or other way of treating it that might be curative. And the problem we have right now is that we detect cancers too late when it's a large fraction stage 4 and it has metastasized to different parts of the body, and so therefore we can't cure it anymore. And so the holy grail then is to come up with a prognostic test that's not non-invasive, so it doesn't involve all kinds of invasive procedures. That could be based on a blood draw that would tell you specifically whether you have a tumor or not, and also where it is. And so there's a company that's called Gray, because this is the holy grail, that has raised $1 billion to try to answer this question. And so we started becoming interested in this when we joined as a second faculty appointment the stem cell department at Harvard. It's part of the college. And these guys have very different expertise than we do. So we needed to learn all of the principles of developmental biology. But I now know that in early mouse embryonic development, at the 8-cell stage, there's really no differential methylation across the genome. So we're looking at a fraction of methylated CpGs across how much methylation there is. On embryonic day 3.5, you can start to differentiate tissues that become the baby. At that point, it's called the inner cell mass, and what becomes the placenta. At that point, it's called the trophectoderm. And it starts to be differentially methylated across the genome, but not differentially methylated between those two tissues. And then as of 6.5 days post-fertilization, at that point, it's called the extraembryonic epiblast ectoderm versus the epiblast C, I'm still confused. And then they're differentially methylated. And they're interestingly differentially methylated. They're differentially methylated in very similar ways in which we see tumor tissues differentially methylated from normal tissue. And we also realized, as we looked at this, that you could just come up with a signature that then defines CpGs that are hypermethylated in the extraembryonic in the EXE, whatever it was called, extraembryonic ectoderm, yes, versus the epiblast, and it's about 200,000 sites. And the patterns, if you just look around the transcription start site, so the site where a gene starts to be expressed, it looks very similar to cancer patterns. So in cancer also, it's a global hypomethylation compared to normal tissues, with very specific hypermethylation around the transcription start sites. So these patterns look very similar. So we then took those 200,000 CpGs, lifted them over from the mouse genome to the human genome, and just compared expression patterns in everything we could find on the internet, which is about 30,000 samples, more or less. And consistently across all of those samples, we found that those EXE hypermethylated CpGs are very consistently more methylated in tumor as compared to the normal tissue. So these are matched normals. For a breast tumor would be adjacent normal breast. And then we look at roadmap and encode, and we also classified our samples according to their methylation status in those lifted over. It's about 868 CpG islands. And we see that they are very significantly more methylated in disease samples as compared to normal samples, with some notable exceptions, for instance, a placenta cell line. And also normal colon seems to be on the higher end of methylation, but not all of them. There are a bunch of colons over here also. So that's interesting because it suggests that we should be able to use this as a blood test. But of course that's not helpful, because if you know you have a tumor, but you don't know which tumor it is, and you can't see it with any other imaging platform, then that's nothing but stressful. So we figured out that we would have to also predict what the cell type of origin is. So of course methylation is very cell type specific, and we can then use a subset of those CpG sites to identify, or basically to align them to known methylation states based on basically roadmap and encode data that we've learned. And so this shows sensitivity in blue and specificity in red across all of the samples that we've looked at that also have matched normal, in order to identify how well we could predict the cell type of origin. And so overall, I think the reason we're so excited about this is for two reasons, right? Number one, we always think about tumorigenesis as a stochastic accumulation of individual mutations. If you accumulate the right type and the right number, then maybe that gives rise to uncontrolled proliferation and therefore a tumor. But this would suggest that there is some kind of a predetermined developmental pathway that was evolutionarily innovated because we're mammals and we need a placenta. That's the safest way to deliver a baby. But it also makes us vulnerable if we switch to this developmental pathway in the wrong circumstances, in a cell that's not a placenta, it's not supposed to become a placenta, that it actually becomes a tumor. And so maybe we are thinking about, maybe there's some kind of a predetermined developmental pathway that if we switch to this, that actually predisposes us to cancer. And it doesn't, of course, preclude them accumulating other mutations, right? But it could be some kind of a switch that then enables us to accumulate mutations. And the second thing, the second reason I'm excited about it is because the dream scenario is we go to the doctor once a year, we get a blood draw, right? We run this panel, and so we're working now on a very deep sequencing panel of just informative sites with our collaborators at Broad, that will then tell us, do we have a tumor and with which probability, and also what's the cell type of origin. And then of course, eventually, the goal would be to try to figure out, can we come up with a molecularly targeted approach to actually stop it, or maybe even revert it, right? And also, then maybe monitor it and find a sub-signature as we accumulate enough data that would even tell us what the right treatment or the right prognostic information would be, such that we don't have the same problem as we have with mammography, right? Which is kind of everybody who has a positive result gets treated, but not everybody who has a positive result needs to be treated, right? So we will have a huge overdiagnosis issue here as well. But maybe there's some more information in those sites that we can actually learn as we accumulate more data.
Any questions for Franziska?
No questions? So even was super clear.
The previous slide, really impressive. Both the sensitivity and specificities are almost close to one. That's really, really impressive. Can you give some intuition on why this works so magically well?
Yeah, it's because it's DNA methylation. That just defines a subtype. And if you look at the right sites, depending on how large your signature is, right? So that works well, I'm not too surprised. The question is, how small can we make the area of interest that we have to sequence really deeply, particularly where we get to cell-free DNA, where we have to sequence maybe at, I would have to see, right? For stage 1 breast cancer, the fraction of tumor DNA in cell-free, so in plasma, is about 1 to 10%. We did some simulation studies with published cell-free DNA methylation reads that show that we can go down to 0.01% cell-free DNA. We can still detect it, but then we already have to sequence like 50,000 X. So, I mean, it becomes expensive, but the end goal then is to kind of reduce the search space enough so that we can still learn things. And the other thing that maybe would decrease it a little bit, right? So we've grouped some tumors together. For instance, there's I think 2 different tumor types. And we thought it doesn't really matter whether it's a, whatever they're called, squamous eye tumor versus ovular eye tumor, right? As long as we know it's in the eye. But if you want like a specific different molecular subtype, then maybe the predictive accuracy would actually go down. But this is all on publicly available data, right? It's all TCGA. So now we just got access to 150 primary samplers from collaborators at Dana-Farber. And we're just going to really deeply sequence them in those sites that are informative. And then maybe we can even reduce the search space and then start a prospective kind of acquisition of cell-free DNA and stuff. See how that goes.
Any more questions? All right, so now I'm going to hand it over to the next presenter is Michael Hughes.
So, most of you know, or many of you know, that my collaborative research is infectious diseases with a focus on clinical trials. So what I thought I'd do today is talk about two study design questions which are very current. They're coming up in clinical trials that I'm involved in at the moment, and we don't yet have the answers for them. So the first trial is in hospitalized patients with influenza. And the idea here is to randomize them to receive a plasma infusion. And the plasma is either a high titer, so it has a lot of antibodies to influenza, or very low titer, meaning it has very few or no antibodies to influenza. And the aim is to try and improve dramatically the outcome for these patients, some of whom will die, but some of whom will actually leave hospital within a week and hopefully resume normal activities. So the design question actually is a redesign question. So this is an ongoing trial. And just by way of background, it has stopping guidelines for efficacy and futility. A very strict stopping guideline for efficacy. So we stop if a nominal p-value comparing randomized arms is extremely small. And conversely, if it seems that it's unlikely that we'll show a difference if we continue to the planned sample size, then we would stop this study based on futility. So the question that's come up in this study is, what do you do in a trial of a seasonal infection when the sample size goal is not quite achieved at the end of a season? And the underlying issue here is that these clinical trials are extremely expensive to conduct, and actually the cost of maintaining a clinical trial infrastructure from one season, 1 year, to the next flu season is extremely high and actually dominates any cost of actually enrolling individual patients. So some options being considered, one is follow the pre-specified guidelines and go to another season and incur a very high cost to enroll a few patients. A second option is just to stop now, analyze the data and accept that you're going to lose some precision and power. Or a third option, which is quite attractive to the sponsor, is to continue for another whole season, so increase the sample size and increase the power and precision to evaluate the difference. So there are a couple of related design changes which the sponsor is considering, and these cause a lot of statistical issues. So one proposal is, can we relax the efficacy stopping guideline for the efficacy analysis at the end of the current season? So in other words, use a different threshold for determining significance. And the problem there is obviously control of type 1 error rate and how to do that in a valid way. And it's complicated by the ordinal clinical outcome that's being used in this trial. And the other sponsor question is, can we change the threshold for the conditional power, so the futility guideline, and only go forward into the next season if there's a very high chance of ending the study with a significant difference? So the question that's being raised to us and to you is, what would you recommend in these circumstances? And it's not an easy question to actually address. The second example is a cluster randomized TB prevention trial. And here the concept is to try and prevent transmission of TB from a drug-resistant TB case in a household to contacts within the household. And the actual clusters of interest are so-called high-risk contacts. These are people at high risk of getting transmitted TB that actually develops into active or symptomatic TB. So it's a cluster randomized trial. We're going to randomize clusters to 6 months of preventive treatment and experimental treatment to auto control, and then follow them for the development of active TB over a 2-year period. So the actual design question, the design of the trial itself is very complex. The design question that we're now facing is, how do you design a pharmacokinetic study to look at drug levels in participants in the study, nested just in the experimental treatment arm? And this has two purposes. One is to, and this is a very big problem in prevention trials, one is to check whether participants are adhering to the regimen or whether there's gross non-adherence, which would make the trial futile to continue. And the second one, which is a little bit more subtle, but it's an extension of the first purpose, is to evaluate the association between drug levels and the development of active TB as the study's going on. And this is really to check that the dosing of the drug that you're using, you've actually got that right in the prevention setting. So it seems like a simple study. We collect blood samples on all participants in that arm, test for drug levels in all the active TB cases that develop during follow-up. And then the concept is to test only a small subsample of controls, so the people that don't develop TB during follow-up. And that's purely a cost issue again. So you can see cost is a big issue in these trials. This sort of study costs something of the order of $60, $70 million, just to give you some context. So it's a very simple question, but it turns out actually to be quite complex. How do you select the controls for the PK testing? So for example, do you test a random sample of controls, ignoring the clustering? Or do you test everyone in a random sample of clusters? Do you stratify the sampling by risk group? Do you test everyone in a cluster, which includes a case? Is there a lot of information in those particular clusters? So the reason this is complicated is obviously the clustering, not only of the outcome, TB outcome within households, but also the fact that adherence and hence drug levels are actually also likely to cluster within households. So you're likely to get households which are very adherent and other households which are very non-adherent. So again, what would you recommend? Thank you.
Questions from Michael? Michelle.
For the flu trial, I was wondering, lots of times we hear about how the flu evolved from season to season. I'm just wondering if there's a problem with no longer comparing apples to apples if you do wait till the next season to analyze the data.
Yeah, so that's a very important question. What they're doing is they're infusing plasma samples from people that have recovered from flu. So the practical challenge is actually getting enough samples early in a flu season that you're getting antibodies to the strain of flu that's actually circulating in that season. So it is a very important practical point. Even beyond that, there could still be variability in how effective the concept is from season to season. This is convalescent plasma. That's rather like what they used for Ebola patients when they came back to the States. That's one of the treatments they tested. It's actually not been well studied in randomized trials, despite its use for decades. Judith?
I think I'm reading on futility analysis, and it seems like you can stop for futility whenever you like, as long as you don't change the final analysis, right? Because it won't increase your type 1 error in any way. It will only decrease it a little bit.
That's absolutely right, yeah. So the type 1 error rate issue really was about modifying the stopping guideline at the interim analysis at the end of the current flu season. So that's the one that would impact the type 1 error rate.
Any more questions? Okay, I only have one question. What type of PQ models are you using? Like compartmental pharmacokinetic models, or?
Okay. So the particular drug that they're using in this study has a very long half-life. So you can do sparse sampling, just in other words, just get a blood sample on any particular day, and there won't be much difference in the drug level depending on when you time that sample. So you don't actually need to get into the types of models that you're talking about.
Okay. Paige.
I just wanted to make a comment. I think it's really nice that instead of telling us what you know, you're asking us. Flipped 5-minute lecture. So I just
Good student projects here.
So I just want to mention, Michael keeps asking, are there any students who don't have a summer project yet? I could really use some help. So if there's anyone who wants to work with Michael on any of these questions.
PhD or master students,
Anyone.
shoot me an email.
All right, and then next presenter is Xihong. And I guess right now would be a good time to get some refills.
Okay, Okay, so I'll share with you what I do, and in particular, so my research has primarily focused on developing and applying skillful statistical inference method for analyzing the massive genome, exposome, and genome data. So one good example of the genome is whole genome sequencing. So I'll tell you the current status of the whole genome sequencing effort in the US and how we are involved. A good example of the exposome is the different type of exposure data measured from all different kind of devices, and from the smartphone technology to the traditional questionnaires to wearable devices. A good example of the phenome is the electronic medical record, which can measure the whole medical history of each patient. So let's start from the whole genome sequencing. And so for each human subject, each of us, we have 3 billion base pairs. This is 3 billion letters across the genome. So each of us have basically 3 billion data point across the genome, ACTG, with all kind of combinations. And so you can see the data, each person's genome will be large. And so here is the timeline of the whole genome sequencing effort in US in the last 10 years. So it's about 10 years ago that was the launch of 1,000 genome project. So that means 1,000 people were sequenced for the whole genome sequencing, but the depth was pretty low at 4x because of cost. And about in the last 3 years, there were 2 national effort, whole genome sequencing effort. One is called the TopMed, this is called the Transomic Precision Medicine Program, and by NHLBI, Heart Lung Institute. And so the plan is to do whole genome sequencing of 120,000 to 150,000 people. And 2 years ago, that was the launch of the genome sequencing program that is by NHGRI. So this was the NHGRI originally launched the Human Genome Project about 15 years ago. And so the plan is to do whole genome sequencing of 200,000 people. By combining these two, that basically will be the largest in the world with the whole genome sequencing data. So we are involved in both of them. And so here's the current status of the TopMed and GSP. And the TopMed has about 40 projects, and then it right now has finished about whole genome sequencing about 100,000 people. And with 100,000 people, there are totally about 500 million variants. So you can think about the data as 100,000 rows and 500 million columns. And so GSP started 1 year later, and so our department is one of the 3 analysis centers of the GSP. And so far for it has finished sequencing about 100,000 people as well, a little half of whole genome sequencing, less than half, so whole exome sequencing. So for phase 1 data, so we have about 22,000 people, and then totally about 260 million variants. So you can think about 260 million columns. And so the first research area that my group does is the analysis of whole genome sequencing data. And so the major challenge here is the signal detection. Most biological signals are weak and sparse. And so how can we scan the genome to identify the genetic variants or the genes that are associated with human diseases? And then this can be used as a drug target. So this will statistically, this will involve a high-dimensional hypothesis testing problem, and to test for the high-dimensional dense and sparse alternative. This is quite a challenging problem. And also, how can one analyze the multiple phenotype, that basically multiple outcome, and look for pleiotropies? So for example, whether one genetic mutation could be associated with the blindness, or the liver disease, or cardiovascular disease. If that is the case, then the one drug can be used to treat multiple diseases. And so this is called the PHEWAS, phenol-wide association study. The other goal is to do risk prediction. How can one use the genome data plus exposure data, like epidemiological data, and to predict the disease risk? And that can help with the intervention and prevention strategies. So the second area is the integrative analysis of different type and different sources of data. So examples include the genetics data, such as whole genome sequencing data, or genomics data, such as the expression data, or methylation data, or RNA-seq data, and like ENCODE, Franziska mentioned, or GTAG or TCGA, but they are collected from different sources, not from the same subjects. And also the exposure data and or electronic medical record. And so to do this, so the way we do it to use the high dimensional causal mediation analysis framework. And so the type of technology for that, it seems like also related to the technology one can use for reproducibility and replicability. And there's one National Academy study and on this reproducibility and replicabilities. And the third area is analysis biobank. Biobank is really exciting areas right now, and also has a lot of promises. And so basically every institution has their own biobank, and also at the national level and also international level. So one great thing about the biobank is has measures the genome and also EMR, like electronic medical record with a large number of disease outcomes, and also epidemiological data, and also like imaging data and wearable devices. Here are a few examples. A good one is a UK Biobank. Some of you probably, anybody heard about UK Biobank? Yeah, great. And for the UK Biobank, it has 500,000 people and has all those type of data. And so it right now has exome GWAS data, but they are also getting exome sequencing data. And also for the EMR has about over 2,000 different diseases, and also has the imaging data and also physical activity wearable devices. And there are many other national and international biobanks, like the China Kazuri or Finland Biobank or Million Veteran Program, and the Geisinger MyCode or eMERGE. eMERGE is the US consortium of electronic medical record. Combining all of those are over 3 million people right now. And another good one is the ALVAS. Anybody heard about ALVAS? Yeah, ALVAS is called the Precision Medicine Program. Probably many of you heard Precision Medicine, right? Program that was launched by Obama's administration. And it changed to ALVAS. The name is changed. So this is the biobank, the way you look. You can look, if that picture looks like a warehouse, right? And so this is how the modern biobank looks like. It's very, very robotic. And so one can basically put a blood sample or whatever tissue sample there, and then everything is digitalized, and then one can pick the right sample from this kind of biobank warehouse. And so all of us finished about 25,000 in the pilot studies, and so just last Sunday, that was a national launch of the national enrollment. So it was launched by Francis Collin, who's the director of NIH, and so there are many, many sites, and so the launch events happen in multiple sites. So with all of those, the goal is to develop precision health strategies that include the precision prevention, precision medicine, to identify the prevention, intervention, and treatment strategy targeted towards each individual person. Hopefully with that effort and the intervention, prevention, and risk prediction or treatment strategy can be more successful. Thank you.
Any questions for Xihong?
Paige.
So I think there's so many different challenging problems that emerge statistically in the inference of this data. How do you prioritize what to work on first?
That's a very, very good question. I think that is very much driven by problems, what kind of scientific question we want to answer, and then set up a priority. So for example, for the whole genome sequencing project, and so right now, yes, we try to identify the genetic region or the construct or the genes, and which are associated with human diseases. So right now, basically, a signal detection problem. But so this is the first priority right now, but there are other priorities as well.
How precise is genome mapping? So if we have hundreds of millions or billions of pairs, how many of those is the process getting right?
Depends on how deep the sequencing is. So in 1,000 genome is about 4x. 4x is pretty shallow because at that time was too expensive. I think sequencing each person, and so the sequencing each person was really, really expensive at that time. And so right now, so for both the GSP top med and the average sequencing depth is about top med is about 38x, and so GSP is about 31x. Franziska mentioned that 5,000x that would be much more expensive. But generally for the tissues, and one want to go a little bit deeper. But those are germline, so generally for 30x the accuracy is really good. So one doesn't need to go too deeper. And so that is a balance one need to make, because if we make the sequencing deeper, and generally it would be more expensive, one will sequence less people. But if one makes the sequencing not deep enough, it's not accurate enough. So this is a balance one has to make. Yeah.
Any more questions? All right. Paige will be presenting next.
All right, so having to give my talk after the chair of the department and my husband is sort of a challenge. I don't know which is worse, but like them, I found one topic is not enough for 5 minutes. So I'm talking about two different areas that I've worked in, both in terms of collaborative research and statistical methods development. So the first area is looking at the effects of endocrine disrupting chemicals on outcomes of assisted reproductive technology, or ART. Endocrine disrupting chemicals include classes of chemicals like phthalates, parabens, flame retardants, and virtually everyone in the US is exposed to them. And because they mimic normal hormones, they can interfere with hormonal pathways and affect our reproductive outcomes. So some of the statistical issues we've discussed here are dealing with a complex data structure, which I'll show in a minute, looking at mixtures of some of these environmental exposures, and mediation analysis to better understand the biological pathways. The second area I'm going to talk about is a different kind of ART, that's antiretroviral treatment. And here I'd like to talk about some safety studies that I've designed and conducted to look at ART and outcomes in children born to HIV-infected women. And again, some of the statistical issues share some of the same features as this first area. So both of these concepts relate to the idea of DOHAD, which you might have heard of. This stems from Barker's hypothesis. And here in the center, we have sort of the Goldilocks of just right, where women are healthy, they eat normally, they have the right diet, they gain the right amount of weight, and that leads to a healthy baby and healthy adult. But at either end of the spectrum, we can have not enough or too much, and that can lead to fetal programming that can lead to down-term effects, long-term effects on adult outcomes, primarily cardiovascular, which you see there in some of the health outcomes. And so because the gestational period is so sensitive, we think this is a key area that could be affected by HIV, antiretroviral exposures, and also environmental exposures. So IVF studies are one way that we can address that, and they're really unique opportunity because we can look at environmental exposures and their association with outcomes that most women wouldn't even be able to know they were pregnant at the time we can observe some of these outcomes. So the key thing to notice here is there's a sequence of events that occurs in an IVF cycle, and this IVF cycle requires that women go through stages, and they have to succeed at a previous stage to be able to continue to the next stage. And it's a combination of different types of outcomes throughout the stages of an IVF cycle. And this is even more clear when we get to some of the later clinical outcomes in an IVF cycle, where we have implantation, that's the usual pregnancy test where you pee on the stick. Clinical pregnancy was detection of the heartbeat, and live birth. And there can be failures anyway along this pathway. And so a woman can only proceed to the next stage if she succeeded at the prior stage. And it's also complicated by the fact that women often have to have multiple IVF cycles before they succeed in having a live birth. So a few years ago, I worked with a postdoc, Arnab Mehdi, on developing an approach that's a discrete survival approach. And this approach
allows different effects of exposure at each stage of the IVF cycle, and still accounts for the shared frailty that women may undergo multiple IVF cycles before they succeed in having a live birth. And so here at the bottom, I've shown some of the adjusted odds ratios, and not surprisingly, here the effective exposure is just their age group. And the oldest women have over 3-fold higher odds of not achieving a healthy pregnancy by the end of the IVF cycle. So this is really a cumulative probability over all of those different stages. And we saw similar effects with an alternative model that was a transitional model. But the main advantage of this model is it allows these different effects of exposure at each stage. And what we found here is that although older women had increased risk of all types of failures, they were especially sensitive to the latest stage of the IVF cycle, which was spontaneous abortions. So other types of exposures like phthalates or flame retardants, they may actually have more of an effect on these early outcomes that we wouldn't usually get to see in naturally conceiving couples. Now I'm going to turn to a different type of ART. So now instead of assisted reproductive technology, I'm talking about antiretroviral treatment. And here I want to talk about some studies that I've designed to look at the safety of antiretroviral treatment in HIV women who are pregnant. And we look at the outcomes in the children, and we also look at the adverse pregnancy outcomes that these women have. So this study called SMART, I've designed, this is a study that's being done by the FACS cohort that I lead with George Siege. And so far we have about 4,000 mother-child pairs. And so I just want to mention the key challenge here is that we're trying to look at toxicity of individual drugs, but all women receive combinations of drugs. And the standard approach is to look at them one at a time, but we know that that can be biased, that can lead to biased estimates. At the same time, it's hard to put 25 individual binary indicators in a model when they're highly correlated. And so here I worked with a doctoral student, Kat Correa. This was part of her thesis. And we developed an approach where each individual drug could be expressed as a drug class mean plus an individual random deviation. So this is a hierarchical model, but not clustering people within a subgroup, but rather drugs within a drug class. And this model actually seemed to have some very nice properties in terms of convergence. It actually converged all the time, whereas the model, if you try to put in 20 different individual drugs, barely converged. It also has lower type 1 error rate, and usually tends to have higher power for detecting true effects. So I'll end with this one figure from Kat's paper where she showed that in the stars here, the hierarchical model tended to have lower false positives, detection of some adverse effect when there really was none in her simulation studies. This was true both for binary outcomes and continuous outcomes. And these different scenarios here represent cases like when there really was a similar effect within a drug class, or even in cases where two drugs had opposite effects within a drug class, it still seemed to perform just as well as a model that had all of the effects in a single model or separate evaluation of each drug. Okay, I'm done.
Questions for Paige?
I'm just wondering if there is a problem with one of the drug classes. Are there enough, I'm not that familiar anymore with the ARV therapies and whatnot, but are there enough that you could provide some sort of replacement for that? You know, what do you do?
Right, so I mean, I think that's the premise here. I think these are the three drug classes, the three main drug classes. There's a new class called integrase inhibitors, but all women receive a combination of drugs, and all of the current combinations tend to be pretty effective at suppressing viral load. So women actually have a lot of choice, and in some sub-Saharan African countries where it's like rolled out as a national program, all women get one of 2 or 3 choices. In the US, there's hundreds of different combinations, and they really depend on prescriber preference. And so if there are options for women to take lots of different combinations that all will be effective in preventing transmission to her baby, we want to make sure that the safest drugs are used. So we do actually do a lot of monitoring to look overall at drug classes, but even within a drug class, I think we're concerned that there could be some drugs that don't actually act similarly to other drugs within that class. So the mechanism might be the same, but there's some evidence that even within a drug class, some drugs might actually not be as good as others.
Last lightning